Hi, everybody. Welcome to the Roach Crossing Weekly General Live Stream, February 2nd, 2022. I just noticed for the first time in all these months that corner is particularly blinding, and I'm not sure if I'll have to go back and look at another video. I'm not sure if it was always like that because I used to have some mesh cages over there that may have uh, helped absorb the light and not reflect it as much as the wall. So I'll ask when we have some more people in here too, but to see if that is particularly distracting to anybody, because now that I've noticed it, I feel like I can't stop noticing it. So we'll have a brief back and forth in the chat about that in a little bit. Uh, Things happening. First and foremost, uh, unfortunate happenings in that this was one of my largest shipping weeks. I had been postponing shipping due to temperatures and took a week off to sort of catch up on stuff, which was wonderful. But this week, once you pointed out, became more distracting, but it's not bad. Yeah, I'll look into doing something, or maybe putting something over that area when I do future streams, maybe next week. So good to see everybody in the chat. Evan G, hello to you too. Jay Brooks, nice to see you. And of course, TJ, good to see you too. We have a lot of things to discuss this week. I'm not quite as peppy as I was last week, not because I'm in a bad mood or anything. I do have a bit of lingering worrying about the boxes that I sent out this week, which everything's getting delayed across the board. But I do pack for that and hello, Alan, had some boxes, or had at least one box back in mid-January that went to somewhere really cold. I think it was Minnesota or something like that, priority mail, and it was delayed like 5 or 6 days and had some tropical mantids and stuff in it, and it was actually fine. So I'm not too concerned, but I can't be happy until boxes reach their destinations. So I just have to deal with that for now. On the list of things this week, it's one of those days where we didn't really have much going on this week, especially with the shipping. But things have just sort of really come out of nowhere with respect to stuff having babies and popping up and blah 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 blah. And yeah, and I actually haven't been receiving any live stuff. Not entirely because of the fact that I spent way too much money on bugs in December and November, but also it's just not the best time of year to be shipping stuff around. Although I had a couple things coming in January, anyways. But right now with the snowstorms and everything, it's still not a good idea. So also might notice that the door is closed. Maybe that also factors into how bright the light is. That's because I am currently eating some very delicious cheese curds, and Victor absolutely loves these, and he would be pestering the life out of me if he realized that these were here within mouth reach of him. So I'm going to keep the door closed until I'm done eating, because I actually haven't eaten much at all today yet. So, things to talk about. I'm going to bring this up at the end of the stream as well, but next week's stream will not be a weekly general live stream. Next week's stream will be a informative, sort of more formal, laid out stream where I will touch on a topic that I feel really needs to be addressed with. It's going to be mostly about. It's going to be. I must have seen a deer or something in the yard of the neighbor's yard, or somebody's running their snowblower and he thinks it's something he must defend the house against. But mostly going to be focused on not going to be isopod lines. that is a whole can of worms for another day. And I think before I touch on isopod lines and like a really, really hardcore, he knows about the cheese. Um, Before I touch on isopod lines in a really hardcore way, I'd like to probably have a better camera system so that I could actually like show things in containers and really go in depth with that sort of stuff. Because I feel like that will make a big difference with sort of explaining and going into that. So that's a ways off. I am looking into bumping up the quality. You know, right now I'm using my laptop's microphone and camera, and I think it would be very very it'll be good for moving into the future with live streams for me to get some more formal gear, maybe a boom mic and stuff like that. Uh, so increase the quality, and I do. This was the gateway into me actually making some content that people can use. It's not as approachable as having conciser videos on different topics, but just getting some general gear so that I can record. Here's how you make this sort of enclosure. Here's this, and doing stuff outside. Here's how you find this. And some of that I think I might be able to do off my phone, but I'm not really going to look into that just yet in terms of using the phone. But the other stuff, maybe getting a better webcam and blah blah blah. So with that being said, next week's live stream will be a bit more focused, and I will probably make a bullet outline. I will still obviously take questions and all the other stuff. There's no reason for me not to. But there's a topic that I would really like to not confront. That's not the right word here. To broach, because it is something that is very near and dear to everybody who keeps any sort of invertebrates, and I think that I could help to shed some light and get into this sort of topic. Anyways, I demand air in a Vega Care documentary so people who are lucky enough to find them can start culturing them, says Alan. Yes. Actually, I was going to talk about my Erna Vega today because I actually went through and did a pretty earnest check on all the ones here, and I have a lot of news. Evan G says if you need editing software, I recommend DaVinci Resolve. It's free and has everything you need. Yeah, looking into free software is sort of my thing. So I would, yeah, I'm gonna. How am I gonna make a note of this? I'm gonna copy it. I have one sticky note on my desktop right now. It's got all the bills I have to pay on it. I'm gonna put that on here so that I remember. So yeah, let's jump in. So I think I brought up in a past stream very recently that I was talking maybe Roach merch. I was talking about telling people that there was going to be Roach merch on the horizon. That is, the gears are turning on that, as well as I want to put up a bounty page on Roach Crossing for things that I want people to I will pay or swap or whatever for people to find. So I'm going to put up a bounty page and see where that can get me, because that I would imagine that some people probably go to my website and see stuff and he doesn't want my this, or he's this kind of guy, he doesn't want this bug that I find or something like that. So that'll be hopefully a better way of getting out the word on some things that I'm looking for that aren't necessarily not evidenced by the fact that I'm roach crossing and blah blah blah. So bounty page, merch page. So on the near horizon, I have contacted the company who produces these hats, which I love the quality. I love the quality. The hat, has withstood a lot of beating over the years. This one's only 4 years old, but I had one before that from the first Arizona trip, and which got lost actually when I went out, not drinking, just went out one night and I left it on the chair because I take my hat off when I eat because whatever, ye old timely respect at the dinner table or whatever. But anyways, that's how I lost my hat. and so then I had to buy another one when I went back. So anyways, I contacted them, and they are willing to work with me to produce a sand roach hat. So now, actually, something that I was in the process of doing tonight, and I may do on the stream, is getting a picture of a pair of sand roaches to put on the hat. I'm not sure if they're going to be able to do two sand roaches. I would love it if we had a male and a female on the hat. I also, if I pick a particularly ornate species, I wouldn't mind having just one big female sand roach across the middle. And so this will be a Roach Crossing exclusive sand roach hat from here. And yes, the hat is size adjustable, and I have a big head, so and apparently whatever happens, that it does loosen up a little bit when I'm working. I could probably tighten this hinges wherever that really was a problem. It never really has been. I mean, you got to tighten up all hats anyways, or put a safety pin or something through it. But they're very good quality. I like to get these when I go to the Southwest as gifts for family and whatnot. And yeah, they are a great hat, and the detail is amazing. It doesn't wear out. Somewhere around here, I made a mock-up hat last year for from this other company. You know, it was just like a big. It was just like a big, make a T-shirt, make a whatever company, and I wanted to try one to see if I could get it anywhere near the quality of this. No, this will end up being. I mean, they it was a certain amount of money for a certain number of colors and all that stuff, and the hat's good quality, but it's just so small, and it's not what I wanted and was hoping for. So I'm not going to go with this company, obviously, because. But anyways, these guys, they make a bunch. The Vinegaroon hat. There's Skull Pendra Heroes hat. There's they're into this sort of stuff. These obscure-ish things, and so it seemed like a good idea for me to approach them about this. And so, unfortunately, that means I'll have to retire this hat. Even though I love this hat so much, I wear it everywhere. But I'll get a swanky Air and a Vega hat. And I wonder if they could be talked into doing blue. I would be over the moon if I could get this hat in blue. Although I will admit, this sort of like camo green is really cool too. And you really can't go wrong with that, with all the as a general for all the invertebrates and animals they have on them, and plants too. They have like Opuntia and Brugmansia, Datura, and whatnot on them. So I was actually going to see if I could find a pair, and this is kind of dumb of me because I did husbandry and I fed things already, and I don't really want to have to go mixing up the substrate layers to dig out a pair to get pictures of, but I would really like to get the ball rolling on this, and it seems like I have a good number of adults in some of these Aranavega containers. So, yeah, I was going to probably pick the locality or species and go from there. I'm deciding between these Cave Creeks, and I have to look at our messages again, TJ. What was it? Apache was the one from Cave Creek Canyon. We figured they were Aranavega Apache. And then from Aranavega from Animas, we thought was Tekka or something like that. Tekta. I'd have to look at the messages again, and I have not committed these to memory because my brain is not like, oh yeah, for sure, that's what this species is. So one of those two, and on the hats for retail, I'll probably just say Aranavega. Maybe if I pester them, they would give the species, but again, we don't have 1,000,000,000% confirmation on that. Yeah, if the Cave Creeks are the dark ones, they are Apache and Tenax, yeah. So I'm trying to decide between the two of these because when we first went to Arizona, and really we hit New Mexico first, the first Aranavega that we found was actually the ones in Animas, which are the likely Tenax. So, to sort of pay tribute to that, I figured, well, that would be a good one to put on the hat. But at the same time, I feel like the one that I really have a deep emotional connection with there is the Cave Creek Canyon ones. So that would be the argument there for the Apache. And the tenax, a lot of Aranavega, especially in captivity, it seems the females tend to become more red. I'm not sure if that's because maybe they sequester more carotenoids from the diet we feed them, or if it's a temperature humidity thing, or if it's a serratia infection thing. So they tend to shift towards red, except for Floridensis, and the Cave Creek Canyon ones tend to stay pretty dark. So I think that would be pretty cool. But then also thinking about how's it going to stand out on the hat, it would be cool because the CF10X are, the adult males are like whitish tan and the females are red, so it'd be very visible. But I do also like the color scheme of the Cave Creek, so I figure that out. And also, depending on which one produces an adult male that I can get a picture of. So let's discuss. I'm gonna put these guys back over here, and let's go in one by one and see how things are doing. So, Aaron Vega from Sahuarita. Where are the Florida ones collected from? Is that sarcasm? Or they are Aranavega floridensis is endemic to Florida, and there's supposedly some formal effort by some researcher or something. Alan probably knows more than I do too. Re-examine the taxonomy of Aranavega floridensis and split it into multiple species. The phylogenetic analysis has already been done. And morphologically, they are very different from each other. So, it's kind of a matter of time and drawing the lines between things. We don't know if they can cross or not. I would think probably, but so that's sort of in the pipeline, is whether those are unique enough to be separate species. And we have 3 species of Floridobolus, and 2 of them are very, very similar to each other, and they've been distinguished based on the isolation of the populations on the different sand ridges. So there's sort of a precedent for things to be split with those distributions. So let's talk about Aranavega floridensis first. I don't actually have them here, but they're actually down there now. So I had good results with Archbold and with Lake Placid, which are very close populations to each other geographically. Could be debated to be the same, population. There's not enough difference between them. They would probably score very closely on that phylogenetic tree. But the people who collected them have been keeping them as 2 separate lines, and I feel like that there's some justification behind that, having been to Lake Placid and then hearing about the distance between there and Archbold. But anyways, so I had good success off the bat with a lot of these floridensis, and then they just sort of start getting die-offs, and they're not quite as vigorous as they were. And so that's been a problem. It seems like it's been kind of a problem across the board with floridensis. And so I had heard from Uncle Fengus, who's the gentleman who sent me the Aranavega booleana from, where are those from? From Bourne, and also Uncle Fengus, if you're watching this, I was waiting for your call last week. I figured something had come up because I didn't receive one, so just shoot me an email and we can plan another time. But anyway, so he has had this colony of booleana going for a while and swears by the amount of oak leaf litter that he provides them as the main reason for their success. And it is my personal theory and probably be confirmed with phylogenetic research, that Boliana and Floridensis are very closely related, and behaviorally they're very similar too, because they're consistently free living sand roaches. They're usually found in leaf. You can still find Boliana in in rodent nests and under refuse and stuff like that. Uh, and I would assume you could probably find Floridensis in gopher tortoise burrows and stuff like that. But people find them pretty reliably free living, eating leaf litter and whatnot. So it was one of those moments where it's like, we should have just been giving them a good amount of leaf litter all along. So, but then I wanted to take a step up from that and was like, the one population in Lake Placid is in like a neighborhood that was supposed to be developed, and then they just sort of stopped. And like at any given time, they could probably. Hello, Hylas Regia. Again, good to see you. I'm just talking about Erinavega floridensis here. But I have a soft spot for Erinavega as I go on and on about them, and I figured I have the space and the resources to sort of do better for them. Having a difficult time propagating them in the first place, is a good sign to re-examine husbandry. So I've set them up in 5.5-gallon tanks, and I have tried to replicate the soil that they're found in the wild to the best of my ability, using the nice soft glacial deposit / beach sand that is a foot down in my backyard, mixed with some high quality sterilized compost / potting soil. So I went all in, and so I have 2 of them set up currently like that. And the one that was sort of, hello, Dynamo Terror, good to see you again. See Boliana. TJ says Boliana have always done very well for me, though, so much easier than Floridensis. My Del Rio Boliana substrate is literally churning with nymphs. Yeah, and my both of my Boliana are now doing well too. So, but they, I'm not exactly sure. And in captivity, things can be different um, from their wild thresholds for things. So while Boliana may be more generalist, just overall, Floridensis, might be something with the heat or humidity or something that we're not sort of touching on, which is why I went all in with these setups in the hopes that I would, giving them enough area to choose from, that I would crack the code and that they would be able to go just exactly where they want to be. So they're set up with nice pulverized, well rotted oak leaf litter. It was actively rotting and rotted. I dried it out, pulverized it, and then mixed it into the surface of the substrate. So I really went all in, because I don't want to lose these, and the collection spots we have for Lake Placid and Tampa, they may be on the chopping block, and who knows when they could decide to develop either of those parcels of land, and then it's a matter of finding another place with them. But it's when you can find them while they're there, and they're going to be gone eventually, why not try? Basically, I wanted to save those populations if I can, so I went all in, and I think that whatever I'm doing is working, because the nymphs overall, or all the individuals, are looking plumper than they were in the old setups. And it could be because they're able to control their humidity better or something like that, or it could be because of the food. Whatever it is, it seems like the conditions are better for them in these enclosures. And I'm waiting on a 5.5-gallon to open up so I can move my Floridensis from Ocala into there. And that's the 3 main branches on the phylogenetic tree for them. There's a couple of other ones I wouldn't mind acquiring, but it's just a matter of getting them. And if you go to the right spot, they can be abundant. If you have a squad helping you look for them, you can get a good number. But the work that it will require to go to some of those other locations and find them, I feel comfortable if I can get the 3 established. That'll be good. And then if I somehow passively acquire other lines of them, then that'll be that. So the other thing I figured was, I'm going to convert this rack into a entirely roach rack. I mean, I am roach crossing, and also having opened up some space downstairs and putting in some new designated racks for things like beetles and millipedes and mantids, etc., will clear up some of the things I've been keeping up here. So, so this rack will have basically a lot of my favorite or finicky roaches. So Supella longipalpa will probably go in a critter keeper up here. Or I have like the Blatella vega up here. The Hemithorsassera vittata will stay up here. And so I will have on the bottom, sort of giving some latent heat to the right there that'll be all the critter keepers eventually. So, but giving some heat to the bottoms of a couple of the critter keepers, there will be a line of all Aranavega enclosure, or not all Aranavega, but probably all sand roaches. So the plan is to have, I have the Tampa there, Lake Placid there. On the end will go the Ocala, and then to keep, because I'm very paranoid, I don't want nails somehow slipping between containers, and they probably cannot get through the mesh on the top of those. But I'm paranoid, and I do not want them getting into each other's enclosures by any means. I have a special way that I make lids, or well, rather, that will make lids. I can make them myself. I just hate doing it because of the meticulousness, and I'm a perfectionist, so everything has to be perfect. So it's easier to have Will do it and just be like, okay, yeah, this is fine. So anyways, if I have to, I'll make those special lids here that I know there's no way that anything can get out of that container of importance. So, but for now, the physical spacing is fine. So anyway, so I'll have the 3 Aranavega floridensis localities, and then the intention from there is this summer I'm going to Yuma to collect. There's a very large Aranavega in Yuma, I would say slightly larger than Bolianum, very similar in size to Polyphaga aegyptiaca, and Yuma is an oven. It's a horrible, horrible place, temperature-wise. In terms of biodiversity, very interesting, beautiful, cool suite of animals that live there. So, but I would like to put my best foot forward, go all in on raising those Aranavega. And so they'll be getting a tank for the heat light as well, so that I can guarantee that I don't have to go out digging around in Yuma again. Because last time, Ben only found a pair of them. So who knows if we'll even find them this time. So, but anyways, I'll be going out there and hopefully find enough to get a colony of those started. And then the 5th container will be reserved for Eremoblata, which probably have very similar long-term husbandry requirements to those Aranavega. And so I assume that they would probably appreciate the heat or the dryness that I'll provide them with the heat lamp. So I'll have a container ready for Eremoblata whenever I find them, or whenever somebody finds them and makes them available again. And that'll be the bottom rung there. And then I'm going to fill in the other containers. I'll have 3 more 10 gallons that will house special roaches. I have the Hemithorsassera vittata. So I'll find 3 other things that I feel deserve extra attention that will go up here. So that's the plan with the Aranavega. Moving on to these other Aranavega containers here. So I thought things were really screwed. I thought we were really screwed in the Animas and the Sahararita containers, because I had bemoaned about those grain mite problems a couple of weeks ago, and it was bad in the Sahararita container. So much so that either the live roaches or roaches that were trying to molt were swarmed by grain mites and basically eaten and hollowed out. Like they were just mummified husks. And it wasn't mismolts or anything like that. Not directly, where you'd see like they'd be partially emerged from the exoskeleton and just dead. No, they were like full intact bodies that were just completely husked out from the grain mites, either the Hypopus or just the regular grain mites that had done that to them. And so very upsetting for me, because I made an effort to collect a ton of those Sahuaritas because they were very easy to find. They were in the sprinkler boxes. So I was upset because I thought that I would have a guaranteed colony of those. Um, and it seemed like it wasn't the case. So I checked on them, pulled out a bunch of bodies. It was horrible seeing all those mummified husks. But fortunately, it seems a lot of the smaller nymphs, which stayed deeper in the substrate to probably avoid desiccation, were unaffected. So there's a good number of medium nymphs, good number of ooths that were unfazed, and so I'm not as worried about I'm not as worried about them. Also, I can go back and collect some more there, or I can bribe Ben to send me more from there. So the Sahuarita are doing okay. Not as good as I would hope, but it's not a total loss collapse. So I'm happy about that. I'm sure they'll rebound from there. Animas, which had grain mites pop up as well, seemed completely unaffected. I didn't find any weird hollowed out husks in there. And there's actually a good number of males and females maturing, and I don't think we found any adults when we collected them. So all of these are nymphs that matured and are getting ready to breed. Hello, Michael Hare. Good to see you. Um, so animas are probably good as well. And I don't see any signs of grain mites in there. It's been very dry in the house, like super dry, because I have to run the furnace constantly because of how cold it is outside. So that probably helps things. And they're all touching a heat pad as well. So animas are doing well. Next up is Deming, New Mexico. This is the place where there was a pile of slag, and it was very hot out, getting late in the day, and I just had to get out because seeing this refuse pile at this rest stop, it was the rest stops in the South and in the West are a lot different than a lot of the ones in the Midwest, where it'll usually be, sort of a grassy, mowed short grass area, a couple of trees. You'll get to like the fence line for the rest stop, and it'll be, disturbed old field, a lot of brome, a lot of goldenrod or something like that, usually butting up to like some agricultural land. When you go in the Southwest and even the South, they'll have things very ornately done around the rest area, but then they'll also have, maybe more intact forest or less disturbed natural habitat, large chunks of it, so you can, areas that are meant for dogs or something like that, so you can actually get out and get to somewhere that's more resembling natural habitat. And then if you get lucky, especially again, a lot of these southwestern ones, because there's just so much space out there, um, they'll have refuse dumps or places they put all the compost or all of the branches or pieces of whatever that they pick up from the rest stop. So there'll be actual spots you can go and find some pretty good stuff. And so this was way back at this one rest stop, was just a pile of concrete slag. So I saw that and thought, Aaron Vega, hopped out of the car, was just muscling through all these big chunks of concrete slag. I was like, Will, you want to help me? And he's like, no. So he just sat in the car, just agonizing, while I was out there sweating and hauling all this rebar and stuff out of the way. And I only found maybe 4 or 5 Aaron Vega nymphs. It's just like, this sucks because this is a new locality and this is all I've got to work with. And so that was the Deming, and I was like, please let something come out of this. Please let something come out of this. And so something has. So I have an adult pair at the same time. The female's been mature for at least a couple weeks. And there's an immature male and there's a mature male. So I'm absolutely certain that we're going to get a pairing. And it's actually kind of easier to start Aranavega colonies with one adult female because she'll lay her ooths. She usually won't eat them. And then, when she passes on, you'll have all these ooths and no other Aranavega in the enclosure to sort of disturb them or possibly eat them when they hatch or before they hatch, etc. Hi, Josh Campos. Hello, Joshua. Sorry, brain just reads off the usernames. Some Egg says in Indiana there's one rest stop with some surprisingly intact forest in a very small woodland. And they sometimes do prairie reconstructions. Yeah, so I would have to say that in my experience going out to like Illinois, Indiana, I haven't actually encountered one of those. But it also depends on what part of the state you're in. I think Wisconsin had a couple of nice rest stops in my memory, but a lot of the time it's brome or goldenrod or phragmites or whatever, and it's usually butting up to ag land, so it's not the average one is nowhere near the quality of one from like from Arizona, where you can like I've done hop out Texas Canyon rest stop and go find skull heroes and all kinds of other stuff. You're also guaranteed to have just one species and then having a mix of species. Yeah, that's another advantage to having just one adult female. So Deming, I think we have a very good chance of getting a colony going from those. Air Vega from Bolin's Old West Trading Post. I want to say that's Las Cruces. Um, I think they were doing so. If anyone recalls, I collected mostly like first through third instars there. Just could not find anything larger to save my life at that spot, and that's also one of my favorite places because of how pretty the nymphs are, very ornately marked. And so they seem to be doing so. I think it's because I had some males mature before females, and so there's just only so many that I could collect at that time. I think maybe I collected between 12 and 20, and then I had the issues with some parasitoid flies emerging from things. So that one kind of up in the air. Fortunately, it's a very accessible place, so somebody else could go and recollect them, etc. But I'm not. We're not in the danger zone there by any means, so I'm not too concerned. Amato has two species. This is from down by the border in Arizona. There's a really good northbound rest stop for bugs. Oh, it's so good. Oh, you could spend just months at that one rest stop. It's so good. So much stuff you can find there. And so this is from pieces of concrete slag that are way out in the hinterlands there. And actually, we've hit those concrete pieces a couple of times now, and the air and Vega always come back. So there's always either booths there. They crawl in from the road burrow section that's close to there, and we do know that there are two species there because there's two very unique female phenotypes, and I think maybe males too. So there are two species in this culture. Just determining which ones, and then of course getting them separated is going to be kind of a pain. But for now, there's a ton of nymphs hatching out in there. There's still a ton of adults, so I have no concerns about getting at least one of those species established. Next up is the Cave Creek Canyon. There was a stowaway Armadillidium vulgare from Cave Creek Canyon in with the Arena Vega that I collected. And apparently it was a female, and apparently it was gravid because they have enumerated immensely. So this tiny culture cup now also has a very dryness-resistant Armadillidium vulgare living in with it. There's probably about 100 in the container starting from that one. I'm not very concerned because, of course, anything that the roaches don't eat, the Armadillidium will eat, which will prevent us from having a grain mite issue in there. So I'm just feeding more. And there's plenty of adult females, plenty of males, etc. So I haven't seen any nymphs pop up yet, but we have so many oots, it's only a matter of time. Some eggs says, so you would say that based on your experience, insect diversity is higher at remnant intact habitats. Asterisk. So it depends on if there's a lot of native species there. Because one of the most biodiverse places that I have ever been to is actually the Interpretive Center at University of Michigan Dearborn, and just the natural area there as well. And it's because there's a mix of habitats, and so you'll have a good variety of species that are found across completely different habitats. And disruption can actually be really good for biodiversity, but again, it depends on whether or not you're getting native species in native plant species and whatnot into those habitats. So a disrupted habitat that has mostly native plants will have very sky high biodiversity, even more so if it abuts a different habitat, and even more so if there is intact habitat in the area, because then things can disperse into the disturbed area and colonize it from having relatively low populations initially. So you want a mix of habitats, you want native vegetation, and you want some disruption in some spots. And my evidence for this is having done the soil invertebrate sampling at U of M as my undergraduate project. And it also, interestingly, you see a higher biodiversity of invasive or introduced species in native habitats or areas that are predominantly native vegetation. So having native vegetation is actually not just beneficial for the native organisms; it's actually beneficial for the for introduced things as well. It would seem. So, and the other thing was a lot of this observation was based on the rain garden outside of the interpretive center, which was once just a lawn, and then they converted it to a very, very diverse native plant. It's not really a garden; I mean, it's a rain garden, but it's not really necessarily directly formally garden. But because of the blend of um, native species they added and the way that things both from the adjacent woods, an intact native habitat, and from the plantings they did, sort of filled in a network. And because of the ecotone of being on a forest edge, while also being a sunny area, while also having a swale where water sort of slows and percolates, has just driven the biodiversity of invertebrates at that site through the roof. So you have other places have a higher degree of endemism, like Arizona obviously is going to have a lot more endemic species because of how things have been isolated by desertification and the specialization and the lack of glaciers in the last tens of thousands of years. But in terms of just sheer biodiversity, which I feel can be more important than just raw endemism at the interpretive center at University of Michigan Dearborn, and the whole natural area there, as the natural area as a whole, it's a much more impressive place to me with respect to appreciating biodiversity, just because the sheer number of things that are there. You see something like differential grasshoppers, where they have a very large range, and you could go anywhere in the United States pretty much and find differential grasshoppers. But then you have a bunch of other widely distributed species there, which means you have a lot of different niches that things can occupy. And so it's not just about, in endemism, things that can only be found just there. It's also about just the high biodiversity overall. It means there's a lot of niches that are being occupied. It's an indicator of very high environmental health to have a high biodiversity at that point, not just the endemism per se. So, Dynamo Terror says, speaking of which, I just ordered a ton of native grass forb seeds to make patch meadows in the forest behind my house. Wonderful. I uh, I am very eager to move to my next property and completely convert it from being what I'm hoping will be an old ag field of corn or soy or something into a verdant native habitat, and one that's also functional too, because I'm going to be raising things like goats and all the poultry and whatnot there too. So yeah, I just I will preach the death of lawns to my death as well. I'm not one to make bold statements, but I'm not one to put my political views out in the open for any reason per se. But the exception is my my extreme vocalism, my extreme amount of being vocal about killing lawns, and so I do have a "kill your lawn" bumper sticker. So it's just you start off, and I'll get back to the Aaron of Vega in a second. But you start off, I guess, looking at the death to lawns, and there is sort of a fanaticism to it where you want to go against the status quo and do something that's going to stick it to the man and to the establishment and whatnot. But once you get past that and you have your established, native gardens and whatnot, or my front yard, although a lot of it looks very untamed and natural, and there's a chaos to it, it is still a garden. I go in and remove invasive species, and I have kept in mind human beauty paradigms and whatnot. But getting past all that, once you see just how much wildlife comes through and uses these areas once you change the vegetation, you really can't go back. People can have a lawn and they'll admire the squirrels and they'll admire the rabbits and all the things that just kind of facultatively use that area. In a lot of cases, because they don't have anything else to use, they're still able to eat things like grass, at least in the case of like rabbits, or they can eat the nuts that fall from the trees that you have in your yard in the case of squirrels. But they're there because they're very generalistic and they're surviving. They're adapting very slowly to these sort of void environments that we've created. And so you can look at them and be like, oh, there's the nature. But then you plant a native planting like this. And then you have things like Carolina wrens nesting outside your window. You have groundhogs making a nice burrow. Fortunately, not somewhere where I walk. But just seeing this and having rabbits raise their babies in a thicket out in your front yard, and the goldfinches and the hummingbirds and the butterflies and the wasps and the bees and the springtails and all this stuff, and you just can't go back. The world seems so empty without that when you don't have the native vegetation that supports all of these things. And you won't get that with a lawn. There's no way you can have all these things with a lawn. You'll have, maybe you'll see a mother rabbit come out, but or she'll make a nest in your lawn somewhere, but it's not the whole picture. They want to nest in like a thicket or something like that, or with like the hummingbirds. You put up a hummingbird feeder, cool. The hummingbird comes and drinks the nectar. What else? It can't make a nest. You don't have the vegetation. You don't have the spiders for them to build their nests out of because they use spiders. So you don't have any of that. They can't feed their babies. There's no insects for them to eat in the lawn. And they don't feed their babies nectar. It's not calorically dense. They have to feed them insects. So they actually feed their babies insects, as do about 95% of our birds in North America when they're nesting. The only one I can think of that doesn't feed its babies insects is, I think, American goldfinches don't. I think that they feed them seeds, but they nest so late, there's plenty of seeds available for them to feed their babies. So, but all the other stuff that nests in the spring, they're feeding them insects. So you don't have the insects to feed them. So people get excited because they basically created a bait to draw in the things that they like without actually supporting them. That thing's not nesting in your yard. It's going to the old abandoned field that's 2 miles away. It's coming out foraging here and found a hummingbird feeder. Of course, it's going to take a drink. So native habitat with a higher turnover rate helps native species establish over invasive. I'm not really sure what turnover rate means here per se, or what you mean by turnover rate. But the idea of having a mix of habitat types, again, is an important factor. You do need to have, for some, so habitat that's managed, and I think they call it a plagioclimax community or something like that, but human managed habitat can be ridiculously productive for wildlife and supporting functional ecosystems. So here in the Midwest, a lot of places will turn to forest, and nowadays things will turn to invasive forest, like buckthorn or honeysuckle or black locust. And so by having fire managed or human activity managed habitats, you can vastly change the trajectory of that habitat's outcome, and also increase its productivity and value to wildlife. So, we don't want to go and destroy every old growth forest because it is a unique habitat. It is performing ecosystem services. There are species that are using only that habitat for nesting or for foraging or whatever. It is a different, you get a different clade of animals and organisms there, and that's important. But usually it's not as high biodiversity as you have in disturbed habitats. So things like forest edges and fields and things like that. And part of that complexity is probably due to sun availability. And of course, the fact that they are changing habitats means that you have a whole different suite of organisms that are exploiting different niches as they come and go. And so that's part of it. But again, we don't want to go and log every old growth forest because there are organisms that are using only that area. So it's important to keep that in mind for management of those sort of things. But in general, these early successional communities do have very high biodiversity, and it's usually, again, you don't have as high of a degree as endemism per se in these early successional communities, but you do tend to have higher biodiversity if they are native habitat. And at least this applies to the Midwest in my experience with that. I love the native species talk, says Evan G, but I have an unrelated question. You have a care guide anywhere for shiny spider beetles? Here's your care guide, Evan. Get a 32-ounce container, put a ventilated lid on it, not big enough that the adult spider beetles can get out. Fill it with dog food, add the spider beetles, and wait about a year. And that is literally all you need to do. I would be hard-pressed to think of an easier thing to propagate with respect to the amount of work you have to do over time and whatnot. And that is my culture of them. It's just I have a larger one because I need more for sales and whatnot. So it's a bigger container than this. But if I threw a dozen spider beetles in here a year from now, it would just be infested with them. I don't have to water, I don't have to change the food or anything like that. It's literally all you have to do. It's just so idiot proof. And I don't like saying that because you can screw things up very easily with very easy species, and then you feel like a big dummy when that happens, and it's happened to all of us. But it's pretty much idiot proof. The only problem I have had myself and some others have had occasionally is when the air humidity increases during the summer, and when the beetles are very active and they, the larvae and whatnot have created sort of a powder layer at the bottom from all their activity, you can have grain mite issues. But if you increase the ventilation and go with a cloth lid, this is, just an example for the illustration. This is actually my red-legged hand beetle culture. But if you increase the ventilation, you can usually, especially if you catch it very on, or if you know in advance, I need to switch my lid. It's summer and the air humidity is shot up, or it's spring and the air humidity is shot up, or whatever has happened, the air humidity has increased. Just switch to a cloth lid, and because the larvae can burrow into the food pieces, and I believe they can actually create water. They can create water that they can utilize within their body by breaking down the carbohydrates. At least I know mealworms for sure can do that. I would not be surprised if other grain pests can do that as well. But they don't need supplemental water or anything like that. Good news. Looks like they will be added to my eventual order from you. Thanks. Yeah, they're so easy. I can't say the same about their wild counterparts that I've encountered in Arizona, but that might be because I threw them in with the Aranavega and sort of let them fend for themselves, which I will not be doing the next time I collect them. I might have to stop by, I think it was Rodeo, New Mexico, to collect more spider beetles now that I bring that up, because I would like to have some wild species and not just the grain pest ones. So, so now, what about those Wisman mealworms? Some eggs. Well, you finish up that thought and let me know what you'd like to know about Kim and Christine's Wisman mealworms. I'm going to continue on with the Aranavega here, and we'll go with next one is Rodeo. Rodeo Aranavega are doing fine as well. I don't remember anything particularly special about the status of the culture. Florida Canyon, I think I found some babies in this Florida Canyon container, so they're doing very well as also. Texas Canyon, this is actually different stock from the Texas Canyon that's been floating around for a couple of years. These ones that I collected now are from the other side of the freeway, across from the rock formation that we usually go and pillage to find the Aranavega Texas Canyon. I do have the original strain of Texas Canyon from the rock formation in the basement, but this is the stock that I collected where I was spooked by that skull pendraheros. So those seem to be doing well. Aranavega San Angelos, from which has been tentatively identified as Tonkawa. I don't think they're Tonkawa. They're too large and they're differently marked, and the nymphs are a different color as well from the Tonkawa I have. It's also possible our Tonkawa are not actually Tonkawa, but I don't know if that's a road to go down or even the right train of thought. So, anyways, I think these San Angelo ones are different species from Tonkawa, so they're doing excellent. Alan was having problems getting them to produce any babies, and he sent them to me. And now I have subadult males, and I think it's been maybe 9 months since I received them. So I have some subadult males from egg cases they produced. So that's there. Aaron Vega. Tucson are doing okay. I don't think they appreciated my neglect husbandry over the summer. There's probably about 15 nymphs in there from oots that hatch, so I'm not too concerned. I think they're going to be fine. Species Socorro also doing well. Species that a fellow named Chris Chu sent me from California. I have 2 nymphs that are doing phenomenal. The large nymph is a female. I sexed it today. The smaller nymph might be male, and if it is, I'm in the clear. I can screw up and recover with just a pair. So if not, then it's going to be very expensive for me to get some more of them because he has to go to a very specific place at a very specific time, and it's out of the way, and it's going to cost a lot of money, but it might be worth it. So I'm just going to really hope that big one or the small one ends up being a male, because then it'll be perfect, because the female will mature first and the male will mate her, and it'll be great. Oh, it'll be so great. Last 2 up here are Monahans, which is another one, again, I collected a lot of, and I feel like I should have more from them, and I just don't. But they're doing fine. Again, I haven't really seen any babies hatch, but there's plenty of mixed size individuals, so that's good. And Fort Stockton, from where I went off at the Shell station and went off just screwing around in the creosote and trash fields, and found 1 adult female underneath it was a piece of newspaper or a piece of cardboard that was stuck on the ground. There are nymphs. I have had newborns pop up as of today, so I think we're in the clear there, and we'll have that locality established just by only starting with 1 female. I'm going to take a look at the chat and see if anything needs to be acknowledged. Have you tried selling Yoni mobius and other Triggs ground crickets? I have a local species that I bred several times. Funny you should mention that. There is a species of Unimobius, and actually, I think from the calls I'm hearing, there might be a couple of species or genera. But the primary one is, and I wrote a blog article on this a while ago. The answer is yes. They are not really a pest. Unimobius carolinus, I think, is the most common one that's in a couple of my bins. Before I had finalized my leaf sterilization, mulch sterilization protocol, etc., they were brought inside and ended up in a couple of my bins and have thrived in a few of them to the point they don't seem to cause any problems for the other organisms in the bins, which is usually roaches. And it's nice to go downstairs and to hear sound in the bug room. I feel like the acoustic experience is very nice. And I feel like there might be a future for people who would like to mix and match to have sort of a sound in their room using different orthopterans. So, yes, I have been propagating Unimobius carolinus completely unintentionally. I actually had Will pull aside a couple of them from one of the bins to start a strictly Unimobius culture so that I can make them available on the website. So trigs, handsome trigs they pop up in the buckthorny areas around here. And actually, well, I had two friends visiting from out of state, and one of them had just learned about the existence of handsome trigs, I think the day before or the day of coming to visit, and then actually found one out in my front yard, which is not really where I would anticipate to find them personally, because reliably I can find them if I go to certain areas that are just all buckthorn. I think that they use it to oviposit, or they prefer to feed on it or whatever. There's no buckthorn. There's no common buckthorn in my front yard, so it must have been feeding on something else or came from a buckthorny patch somewhere in the neighborhood. Um, so I've tried them, not when I had more of an orthopteran mindset, so I wouldn't mind giving them another try this year. And they may end up being like the Tafalisco, where they'll actually eat just fish food. Actually, I'm going to turn off my Air since I haven't actually put them on the timer circuit yet, so I still have to turn them manually on and off every day. I do have, there is some sort of small scaly cricket that I collected in Arizona that I'm actually quite fond of, and they laid a lot of eggs. And I still have an adult alive, actually. I don't know how this one beat up female is still alive. She is from September. It's got a really weird common, or really weird Latin name. I'm gonna see if I can find it on Bug Guide really quick. Arizona red cricket. See if that'll get me anywhere. But I can't remember what specific grouping it belongs to. It's not a sand treader cricket. I would really have to look it up. It's like it's got an H in it. It's like Haplocnemus or something like that. It's a monotypic genus as well. So yeah, I do have some experience with smaller orthopterans, and we'll be making more Grylus available. I see Lewis, welcome. I see your comment. I'll get to it in just a second, since I just brought up Grylus. Inverberju says, now will any of these Aranavega be available this week, even on a single pair basis? I am going to hoard these Aranavega until I have about 100 of each, and then I'll be making them available, just because I want to be extra sure, because I don't want to have to do another 9-day Arizona blitz to recover any of them. And, even though we weren't necessarily hard-pressed to find a good number of each of them, I don't know how conditions will be in the future at some of these locations. Like, I'm not going back to the Deming location, most likely. So, and Sahuarita, I would like to get, up and running again. Alice says, somebody say something. Dynamoterra, I've kept my tiny crickets primarily as a sleeping aid. Yeah, the Unimobius call is very peaceful. It's not like the Grylotes, which I also have, which apparently can get on people's nerves. Some egg says, do North American crickets need a diapause? Yes and no. There's just a couple of years ago, there was a huge, or maybe even more recently, there's a huge overhaul of North American Grylus. And there's a free PDF somewhere online. And all done along lines of locations, and they're finding out that some things which were different species are the same, some things that had different calls are the same, and basically the group is just apparently very quickly speciating or trying to in North America or something like that. So, but there seems to be a genetic basis for splitting species, at least along lines of whether or not the nymphs overwinter or the eggs overwinter is a basic phylogenetic split. And so the fall field cricket, which has eggs that require a diapause, is one entity, and then there's a bunch of them that actually don't require a diapause. at all, and then some that facultatively diapause as nymphs. So the species that I currently have are, it's believed to be a Gryllus firmus from South Florida, which requires no diapause at all, and Gryllus, and I thought they were firmus. There's a mention in that paper of there being some inland populations, but I don't think it's firmus, but I could be wrong, even though I have them labeled as firmus in my colonies currently. Um, and those do not need a diapause as eggs because my eggs just started hatching, cycling through that I had brought back from the Arizona trip. But Gryllus firmus is one of those ones where it seems like some nymphs overwinter, some of them like just grow really fast and breed right after they hatch. So it depends. But the bottom line is the species that I would have available do not seem to require a diapause as a culture. The Gryllus similis are very easy. And I actually have no problems with keeping Grylotes in a large enclosure, the adults, nymphs, and eggs all together. I feed them well, I provide them heat and ventilation. And actually, the more that I work with crickets, the more that I realize how dumb it is to sort of remove the egg cups and do all that stuff. Although there are some merits there if you're trying to get standard incremental sizes for whatever reasons to putting an egg cup and then removing it, etc. You are able to better modulate the sizes you have available, but it's a lot of work to do that. And I don't have the time or the patience to do that. And thus I have my large colonies of things. So I have them propagating all together in one big colony, all life stages without any issues. I have suspicions that over time, without changing the substrate, you might build up some egg pathogens that are just, have that are present in low numbers in the like fungi and whatnot, that are present in low numbers in the substrate. And then over time, the buildup of all the discarded egg cases and dead bodies could affect the vigor of the culture long term. But that's very speculative, and I don't have anything definitive to say that that will happen. I can at least say that most colonies that you set up that way are good for at minimum a year with good husbandry. I also know a field where I can find handsome trigs, so I might try to keep those again. I believe there's an image of a handsome trig ovipositing into a buckthorn branch. And that's another thing with these different orthopterans is figuring out if they lay eggs in wood, if they lay it in soil, if they lay it in branches, if they lay them in leaves. And it's pretty surprising, I would say, finding what does and doesn't do what. For example, the Taphiliska eleuthera, the Florida false ruedas, as I've been calling them, you would think from the information on them and seeing them in the wild, okay, they live on the mangrove leaves, they eat the mangrove leaves, they have a pretty sturdy ovipositor, they probably lay their eggs in wood, and they probably only eat leaves, fresh leaves of things. But then you bring them into captivity, and they're doing just fine on just fish flakes. And from my past breeding experience, failure with them, in that I forgot about the egg cup and it hatched on my counter, and I was like, oh, that's what that is. When I found a nymph that had grown a little bit next to my cuttings, they lay eggs in soil, or at least they will lay eggs in soil. So you got to be a little bold with trying to figure out and experimental with trying to figure out how things prefer to be kept, because it's not always very intuitive. Another example is you look at a Scuduria katydid, and you look at the ovipositor and the shape of it, and then you look at a Stilpnichlora couloniana ovipositor, and you compare the two of them, and you think, oh, these are very similar. They must lay their eggs the same way. And then you find out that Scuduria lay their eggs inside of leaves. They will actually grab the leaf and insert the egg in between the layers of cells. The egg is laid, like, pretend that a leaf is made of multiple sheets, kind of like pieces of paper, really close together. The Scuduria lay their egg in between the sheets of paper. Then the Stilbniclora, which again has a very similar ovipositor, just lays its eggs on the branch, just lays eggs on like a little stick or on a little twig. So there is some observation and experimentation required. So again, the Gryllus similis, very easy. I have a good colony of a couple hundred going right now, and have not had any issues. So, and again, temperature, crickets tend to like to be kept warmer, and I've noticed that they will eat each other if you don't provide a consistent source of food, but not necessarily moisture. A lot of my cultures get sprayed daily to every couple of days at this time of year, and that does not seem to be a big factor in them eating each other when there's some moisture in the substrate. It's more about having just a raw quantity of uh, not necessarily fresh, but not moldy, just available food available. And they have mandibles, so they can chew into it at all sizes. So another sort of not rookie mistake, but another intuitive mistake is you think that things would prefer to eat their food soggy or something like that because it saves them energy, it's more palatable, etc. But when you wet the food and conditions are very moist and there's not a lot of ventilation, it's going to mold, and that's not as palatable to some things like crickets, versus roaches or isopods may chew through it, but crickets will just ignore it. I assume you breed the crickets like roaches on a substrate without incubating eggs separately. Yep, basically the same thing. The substrate's a little different. I have an obsession with mixing sand into orthopteran substrate right now because it's better for drainage, and it's more inert, which I feel is better for eggs. And it varies by species of orthopteran, but I prefer to use some sand in there too. The drainage part also comes in handy with grasshopper eggs, especially in the cups, because the water will drain in and then it will spread pretty rapidly through the other parts of the sand. And it's also easier for them to lay into, and probably more palatable and natural for them when they're ovipositing for certain orthopterans. Some things like the Unimobius are breeding in some containers where the substrate is just turned into basically topsoil with no SAM content. So obviously they don't care very much. But for my Gryllus, I do offer some sand in the mix. Are you still using the egg cups for lubbers? Yes, I am, because to be able to give them a proper diapause and cooling, and also to kind of have some control over when I get hatches. So for lubbers, I do still use egg cups. My assimilis that I got from you are multiplying rapidly. Yeah, that's what I like to hear. I'm kicking myself because there were some very cool Gryllus in Arizona, and I didn't set them up when I got home. I sort of low prioritized it because of just how many I found. And next thing I know, they all died. But it was like, oh, okay, the egg cup's fine. I'll just wait for a hatch from those, like what happened with the Nebraska ones. And apparently, the soil that I grabbed, or the adult crickets, had fluorescing on them, some velvet mites. And so the velvet mites went and ate all the cricket eggs. So that didn't go so well. So I have to collect more Gryllus from there next year or whatever. But also really cool ones that were black with red legs from higher elevations in southeast Arizona. I don't think I was able to catch any females of those, or the female died very soon after catching or something like that while we were on the trip. But I really want those because they're probably something very unique considering their location. So touching on other orthopterans, I do have a good budding collection of Pseudophilus species, or Pseudophilus species and related genera. I have the Gracillipes. I have the Agassizii from TJ, which are doing so right now. I think I'm actually going to bump them to a larger enclosure soon. Pseudophilus CF, I think it was Chiricahuensis, a Chiricahuan endemic Pseudophilus. We seem to have collected from up by the southwest research station up in the Chiricahuas. And the female died very soon after I set them up, and I was like, oh, well, this is, we're screwed here. And then all of a sudden, a bunch of nymphs popped up. So like, Ooh, that's pleasant. Uh, some really cool ones from near Animas and Rodeo that just have this very ornate coloration on them, and it's very hard to find them. They I remember on past trips, maybe a handful of times, seeing them just at the entrance to rodent burrows, but they're so fast and so alert that if you try to approach them at night, they'll just take off. They'll just either crawl down into the burrow, or they'll hop away and hide in some mesquite or creosote. But I found 3 of those on this trip, and unfortunately, it really looks like they're all males, which sucks because they're so cool. Um, but what can you do? So I'll just hope that the smallest one ends up being a female, or that the species has a reduced ovipositor, and we'll see what happens. And then I have a CF, I think it was Meridianalis, that a somebody I went and did some native planting stuff for actually found back in November. It was inside her office, and so I went and or she dropped it off my neighbor, and then he gave her to me. She unfortunately passed away again relatively quickly after I had set her up, maybe within about 10 days. But she was in a very good quality enclosure, so I would not be surprised if there's going to be some babies popping up. And then finally, Kaya and I went and did a little blitz outside to collect some stuff out near my compost, which has a bunch of pieces of wood around it, specifically for going and looking for bugs. And I found a couple of Pseudophilus nymphs out there. You usually only find 1 or 2 at a time, so I haven't really bothered keeping them. But this time I found 3, so I was like, yeah, what the heck? And I have at least 1 pair, and I really actually need to go downstairs and set those up. So I'll probably do that tonight in something more formal. They might be Meridianalis, but I think they're a little bit of a smaller species than that. So Bassoophilis are very cool. I like them. There's just something very quirky about them. The fact that they're completely wingless and they're tritivores, and a lot of them seem to have wonky life cycles where they don't really have a diapause and things like that. So it's a very unique group, and the related genre are also very cool. So touch on Aaron Vega, trip stuff, other things coming ready over here. Roach Dump. Roach Dump is scheduled for this Saturday. Or Sunday? Sunday? This Sunday. So I'm gonna work on it all day Saturday and part of the day Sunday. But Sunday will be the big Roach Dump on the website. I feel like there's a couple of cool curveballs I'll be uploading. I'll spoil some things right now in that Pseudoglomerus magnifica Cuckfuong will be going up for sale. I have several sales of them already, so they may be sold out as they are listed, but I feel pretty confident with their husbandry and with the results that I'm having to make them available. So Pseudoglomerus magnifica will be up for sale. Panacea Angusta penis cognata from Laos will be for sale as well. So that's one that's been pretty coveted over the years, is Panacea. As a roach person, I really like them. It's neat that they're solid black with the orange square on their thorax. They're really cool. I am a little hard pressed to figure out what the sort of craze over them was. When I got into roaches, it was very difficult to find them, but there were some people in the United States who were keeping them sparingly. Well, they're very much so something that people kind of foamed at the mouth over. And I'm a little hard pressed to find that insane enthusiasm for them. They are very cool. They are kind of like sub-social in that there is some nymphal care and sort of not as much as in like Salgania or Ancaudelia, but I mean they're cool roaches. Nice to have a jet black roach with orange on it. And what was the last one I was going to be adding? Something cool. Hemithrisosphera vittata. They are past. I have nymphs that are past 3rd instar. They will be going up on the website, available for sale. I feel very confident in my husbandry and the perpetuation of the culture now that I have possibly even L5s. The first instars just take forever for some reason, especially the first instar seems to take forever. And so that was, of course, my hesitation was I wanted to make sure I could get them past the stage where I had difficulties before, but now I have plenty of them, and so I can finally make them available and declare keeping them a success. And of course, it won't be a 1 billion% success until I have produced F2s, but I feel like we're far past the stage where anything could have gone wrong, and they've survived over both the house being dry and humid and then dry again in this setup. Have you ever tried breeding the big beach flea amphipods like Megalorchestia? I tried once, but my husband was faulty, so they all died within a week or two. I have a colony of beach fleas from Key Largo and some isopods that seem to like saline conditions as well. I've never done exclusively a beach flea enclosure, but I feel like they would do very well in the Aloniscus setup, actually. So if I have the opportunity to try with some of the larger amphipod species, I'll give it a go and do the Aloniscus setup and see how they do that way. I'm supposed to get some Ligia in the semi-nearish future, and I had a lot of good success with Ligia baudiniana, which is found in Florida, and I was able to get immatures to like fourth or fifth instar. And the problem that I encountered with Ligia was that they required constant fresh food. So I couldn't really do the thing where I do with most cultures of stuff where I could just throw in a glob of stuff and, if it gets moldy, whatever, if it gets eaten, I can take my sweet time sort of adding more stuff. The Ligia, when I was feeding fish flakes, and I think I gave them a piece of zucchini at one point too, regularly. So every day I would sprinkle some fish flakes onto this one spot, and then it would be gone by the next day, or there'd just be like one little piece left by the next day. They did phenomenally in the setup that I had them, and I had all kinds of babies. And again, they got to like fourth or fifth instar. And then I can't remember for what reason I stopped doing this daily intensive care for them. When I stopped doing that, within 2 or 3 days, all the babies were not dead. They were all gone. The adults ate all of them. So I had maybe 15 adults, and they got huge. They got like this large. About 15 adults, and then all the babies were gone. And I sort of, after that, I was just sort of like, wow, this sucks. And so I stopped doing sort of regular care with them. And the adult numbers dwindled over I want to say a couple of months. It was a surprising amount of time that they dwindled with suboptimal care. And eventually, I was just down to a couple of really huge ones, and I stopped putting the effort into them. So, yeah, so I would like to raise Ligia again, and maybe I'll do something stupid and get one of those automatic fish feeders and set it up to dump fresh fish flakes in every 24 hours so I don't have to do that, because I'll do it for a while to get the culture established, but that's a lot of work, and how am I going to do that when I'm on vacation? None of my other cultures even need that sort of intensive daily care. They can all be planned. Even orthopterans like grasshoppers, I can just add like a head of lettuce or something, and they'll take care of themselves. So I'll think about it and see what species I can get. Maybe Exotica will be a little bit more forgiving than those Baudinianas were. So, but it's just a matter of also getting them. There was that eBay seller, right around when isopods became really popular. There was that eBay seller who was selling Ligia palassii, and he had those pictures of them on just like wet seaweed or something. It's just like maybe like 50 good sized ones in the middle of this tank. And he was, if you asked him about breeding, oh, they breed very fine. They breed very fine like this. Oh, I have many babies like this. And I was like, something's not right here from my experience and just looking at this enclosure. And then next thing you know, they were just gone. This guy just completely disappeared, and nobody was able to get Ligia Palatii for sale after that. And people were saying that they lost there. So it was like, yeah, those were all wild caughts, most likely. And you didn't have any breeding. You were just keeping them like that until you sold them. So, um, so let's see what TJ has said here. By the way, a little excitement for this year. I might be able to get Paranopha rufipes back into the U.S. hobby. I would be very happy about that. I would be very, very happy if we were to get Paranopha rufipes again. And I feel like enough time has passed and I have enough experience with Paranophita that we would not lose them again collectively. So that's awesome. That would be just amazing if we could get those going again. As I say this, my Formosana, due to the summer neglect/maintenance schedule, had a large crash from being a colony that I could reliably pick 100 out of whenever I felt like it, to being maybe like 20 barely okay condition nymphs and adults. And so I corrected the husbandry, and now we're in that phase where I've seen a couple of females that look very healthy and gravid, but I just don't see enough of them in there for me to be like, oh yeah, this is going to get reestablished and be 100% fine for sure. So I am just a little inkling of concern for them, but I'm probably should just stop thinking about it because they're doing, they recovered so well. And I'm pretty sure I've seen adult males and females at the same time. So things getting mated or not shouldn't be an issue. Oh, also, really quick, I just realized we have the Russian bot again. Hello, Beppa, however the hell you say that. Hello, Russian, the weekly Russian bot. So my Lirata are doing fine. I sent Alex Baranowski some this week in trade. So that's fine. I think Lirata are less common than Formosana, or at least they were. I don't know. Who do you know who has Paranophita Formosana? I almost said Rhabdoblata Formosana there. Is getting vummy in here. Let's see. What else do I say? While the hive mind is figuring out who has Paranophita Formosana, trying to think of other things. Ooh, I have something exciting to show off. Really quick though, I wanted to touch on something else I think I brought up a couple weeks ago. Ty sent me a box with some goodies in it. Oh, TJ says that the reverse is true now. Formosana are kind of rare in the U.S. That's not good. That's very concerning. This is very concerning. But like I said, I'm not very. I just have just a little tickle of concern that I might lose the Formosana. I think we're past that though, and I think I should just stop worrying. The other problem is it's very difficult to know how many Paranophita you have, especially if the setup is larger, because the nymphs just go straight down and they don't seem to stop going. And it's not even so much that they're like Pictoscalus where they'll just burrow straight to the bottom when they're disturbed. They seem to burrow under and then over, so you can never, if you lift a piece of bark that had a bunch of them under it, you have to do a lot of digging around to find all the nymphs that scattered once they scatter. So So I haven't seen any newborns, and once I do, I know the culture will be fine. I have to get the cheese. I'm very hungry. So this thing I was going to touch on, not the thing excited to share per se, although I'm excited to share everything, is I received from Ty in the box of goodies I mentioned earlier was a small group of Pseudoglomerus tarsalis. And I have to say they're doing phenomenally. In fact, they're doing so well that I might move my Paraspherus into this style of setup over here because they seem to be doing so, and they might appreciate having less space, as some things tend to do due to the Elite effect and blah blah blah. So the Pseudoglomerus tarsalis, I believe the largest one was probably a third-ish instar or fourth-ish instar that I received, and I'm pretty sure I have adult females now, and it's been maybe like a month ish or less than that. So they're just doing really well in this setup. And so I wouldn't be too surprised if by April I could offer them, which is, weird and exciting because it took my Magnifica. I want to say I got them 8 to 10 months ago. Could have been sooner than that. It could have been June, but 8 to 10-ish months ago. And only just now do I feel comfortable making some available for sale. So yeah, Tarsalis. I know TJ, you have Tarsalis as well. I may pester you about picking up a couple more. I'm not too concerned about inbreeding, as everybody should know by now. But it's nice with these species that with everything right now, just I like getting large groups of things to start with, just because it means that I can get large established colonies ready for sales and for trades faster, and feel comfortable doing that. Versus if you start with one female of something, okay, you only get so many babies, okay, and then they're going to grow up as a cohort, and you have to wait for the first batch of babies from there, and it just takes a long time overall. Versus if I can get a large group of things, large first set of babies, and I can keep a good colony going without worrying about overselling, etc. So I wouldn't mind getting some more Pseudoglomerus tarsalis. So, but the cool thing to show, it's actually more exciting, I think, than I'm letting off. And I will concede that TJ, you beat me to the punch with getting adults from these. So, but I have double whoa. I have adults of Eleotes spinipes macrura now. The pupation setup, which I feel needs some tweaking, or at least this current iteration did, where I use pretty much exclusively clay. The larvae don't seem to be the best at burrowing into it. They'll do it, and obviously you'll get adults and things will be fine. But it's not working as well as I hoped it would, just the pure clay. So I took a mess of larvae and put them into 50-50 clay sand, about 3 inches of that. And we're going to see if 30 Eleotes spinipes macrura larvae will pupate in a 16 quart shoebox container with each other. So yeah, this is awesome because these things are huge, and these are I will say they are smaller than the wild caughts. Although some of the wild caughts were smaller. I grabbed all the ones that I could find because I wanted to, they were all the same species. I want to make sure I had a good group to bring into captivity. But I think this is a pair. And I asked you on a text, TJ, but that looks like a male right there, right? He's got a mukro, and it looks like he's got an extra spur on the front. What is that? The femur? I don't know if you can see that. See, there's a little spur there, which this one, presumably female, does not have a spur on the front tibia. So this is awesome. And the ones I collected, I've sold a couple of them, and I still have a group of what I think is one male and 2 females of the wild caught ones, because I figured, I'm going to be producing these, and I have plenty of larvae, so I was selling a couple of the wild caught adults. But this is a big, charismatic, easy to care for, easy to breed darkling beetle. Yeah, definitely male and female. So we got a pair right here. And so there's one more female then that emerged in the other container. And I was going to do a temperature thing where I put one cooler and I put one warmer, and then they just ended up pupating anyways. It's just like, okay, well, I'll just leave them at room temperature in the office, then it's fine. So, um, so we have a cool, charismatic, easily captive bred darkling beetle, and I could see these giving blue death feigning beetles a run for their money because blue death feigners obviously are very cute, and they've got the color going for them. But these things can get huge, and now we can get competitive about it. We can see who can produce the largest Eleotes spinipes macrura and go from there. Um, Evan, have a good dinner and have a good rest of your evening. We are at an hour 35 minutes already. Jeez, I don't know if I have some commitments to tend to tonight, so we'll probably go to 2 hours as close as possible. So thanks for joining for as long as you did, and have a good dinner, etc. So, but yeah. Uh, I will hopefully be getting the other subspecies of spinipes in a couple of weeks, pending a couple of things. So, but yeah, these are awesome. And again, I said the wild caught female was larger, but this one here is larger than I think the average size that I collected. So I think that's a good sign that the husbandry that I've been doing is good, and that maybe just grabbing. Do they have any red on them? Nope, they're a nice solid black, and they have a very pleasant texture too. They're not completely smooth, but they've got a lot of very subtle striations on the back. But yeah, some Eleotes do have those markings, which should lead me into, and they're very docile, as you can see too. It just the one male was having a little bit of trouble at first, but I think they've adapted to climbing on my hands, and of course I'm not flailing them around, and they're not freaking out because they got picked up. So uh, have a couple of IDs on Eleote, the other Eleotes I have culturing. The ones from Animas were identified as. I have to look at my emails. Why is this not coming to my? Uh, TJ might snipe me on it. So Andrew Johnson, who's a Eleotes, did some Eleotes reclassification, whatnot. Has been helping with the identification of some of these darklings. Mostly the ones that TJ wasn't very confident on identification with, because this is a difficult thing to do. So the ones from Animas are definitely Eleotes Anthracena. So we have Anthracena in culture now too, and those ones do have a very pleasant red hue across the back on the elytra. Not all of them seem to have it, if memory serves me, and we'll see what happens when captive adults start popping up. But all the adults so far, or most of the adults so far, I think have a very pleasant red. Do they have a red on them? It's kind of hard to tell this. Do they smell? These haven't sprayed me, and pretty much all Eleotes, especially in the wild, if you pick them up, they will gas you or liquid you or whatever exactly the process. Liquid, I think it is. It's a volatile liquid. But these guys didn't, and it might take them a couple days after they mature to build up a stockpile to use. But in my experience, most Eleotes in captivity will not gas you when you pick them up. It seems to be something that they have more triggers to do so when they're in a wild setting. Now, the Paradise Eleotes have been gassing a lot when I pick them. Squirting, I haven't really been hit with a projectile Eleotes squirt. They probably just sort of exude a bit of it if they need to, and they'll commit further to it if they feel like they're really in danger. So, but all of them can do that to the extent of my knowledge. Whether or not they can actually get some distance with it varies by species, it seems. So, but these guys have been very pleasant. So yeah. So Anthracena from the Animas. And Andrew hasn't replied to my notes on the Paradise Eleotes, but it seems their identity was very up in the air. And his hesitation suggests to me that there's a possibility they could be something undescribed. So it would make sense. They're in a very remote location with a high degree of endemism. So they could be something new, but we'll see. So I asked him if he needs a specimen. I'm waiting for him to reply. Who knows if he'll get to it? Maybe I'll have to pester him in a couple of weeks or something. So yeah, Eleotes and the Ambaphian contusum, the one female we found in Las Cruces. Uh, there are now fresh adults emerging in the enclosure. So, and I think I counted maybe somewhere between 12 and 15 of them. So we will have Eleotes contusum in culture again. And I was trying to figure out what subspecies and blah blah they were, but Andrew said that they're just eliminating the subspecies nowadays for the Amphipods contusum. They're just all contusum. And then by extension, I guess would be a contusum subspecies of contusum. But you don't even need to make the distinction if there's only one species without any further um, delineations. So they're Amphipods contusum. So we have both Amphipods in nowadays around, and people. I feel like another thing on my agenda again, once I get a better camera or figure out a better way to do this, is I will go through and I would like to do a communal Eleotes Darkling Beetle video where I, say, here's a setup, here's the different species you can keep together, and they're very active and very easy to accommodate in terms of temperature and humidity and food. And people already love keeping blue death feigning beetles and whatnot, but the setups just are not very conducive to propagating them in colony form. Versus, with how easy it was to get these guys to pupate, that puts them as a pretty good candidate for a communal species in my book. And the larvae did not seem to be particularly predatory. They may have been eating eggs from the adults at high densities, but they definitely were not going after each other. So, and there's also, once you get to a certain density in an enclosure, eggs just get trampled. They just, newborn larvae just can't find anything to eat, etc. So there's sort of there's a holistic effect to look at there. So, but yeah, these, I don't know. I really would like to get the roach dump done in its entirety this Sunday. So I don't know if I'll list these. Maybe I'll do it tomorrow or something. But I feel pretty confident listing these uh, on the website for sale. And I mean, they're just so cool. And I don't remember seeing these the last times that we went to that rest stop. Maybe they were smaller. Maybe conditions were poorer, so they were smaller. But I really don't remember seeing them because I would have wanted to bring back a huge darkling beetle like this, just specifically in Eleotes because I'm familiar with their husbandry. So TJ says, I will say Viricosis seems to be one of the most prolific cryptoglossinae, so it's quite possible yours were literally just subsisting on their younger siblings. I remember having to add more dog food to that enclosure for them. And again, the death knell was when it dried out completely. Those larvae are not very dry tolerant. I mean, Eleotes and Amphipods larvae overall don't seem to be very dry tolerant. I feel like they put all the effort into being desiccation resistant and sort of just stress resistant goes into creating the adults with a lot of darklings, because the larvae are just sort of meant to, as a general trend in my opinion, grow very quickly and get to maturity so that then they can withstand the cold or the poor conditions, etc., as the adults. And again, there's exceptions to every sort of trend, and things like the Suturalis and obscurus overwintering as prepupal larvae may factor in. But then you have the selective pressure of the weather, perhaps, in those sort of populations, where they, for whatever reason, have adapted to do that because it's the best thing for determining when to emerge when good weather has come back to their parts of their range, and I believe both of those are more northern. I remember we found Obscurus in Arizona, but I think there are different subspecies than the ones that are further north. TJ, you'd be the one to know about that. So, but anyways, big, awesome, easy, captive bred darkling beetles. Oh, I just look at my little saints. They're just hanging out. I wonder what will happen if I pet. They don't really seem bothered at all. Oh, they're just so cool. Just look at this. Look at this. Look at these. Look at these this lad and this lass. And since this is a sort of average size adult male, I really wonder if if I get any big males to emerge, if they're going to have a huge mucro too. The little point on the end of the little point at the end of the elytra there is called a mucro. So really, really curious if we'll get some huge ones, and it gets right in the eye. That'd be my luck. And these larvae, I jumped the gun on pulling to get in those setups because in the crappy little shoebox that I had them in, they were getting very overcrowded, and I was having some deaths. I completely husbandry because they were so crowded in food and substrate quality decline and all this other stuff. And so I sort of scrambled to pick an assortment of sizes of larvae to make sure that I got both males and females in case there was a dimorphism in the larvae because of my husbandry at that point. So, but with this set that I threw into that 16 quart container, where I think I picked about 30 big larvae. There were some big larvae in there. Like the head size was maybe, I'd pull up an eye ruler before I sound like an idiot or unimpressive, but the head size on some of these larvae is the largest I think I've seen on any darkling beetles. There were some that had a head width that was pushing a centimeter, definitely 8 millimeters, but some that were maybe even closer to a centimeter, 10 millimeters. And so I'm curious if those will turn to these just monster size adults. And I don't really plan on selecting for size. Maybe over time, if it seems more heritable to me, I will. But I like maintaining things of straight species as close to sort of a natural distribution of size and characters as I can. So I don't tend to cull minors or pull a lot of majors for different reasons. I like to keep the as much of the inherent diversity as possible in my lines. Not in things that are bred for specific traits like the LLE mahogany hissers or my isopods and whatnot. I will cull ruthlessly and hold things to a uh, very specific standard. But for straight stock stuff, I like to keep it with as much variation as possible as it would be occurring in nature, or rather without my specifically guiding hand. Speaking of darklings, isolating as Bolus mexicanus eggs right now, says TJ. Awesome. I think Alan just found his first mexicanus adults, or was pulling some mexicanus adults from ones that he pupated. I would be interested in throwing my hat in the ring with those mexicanus to see if I can uh, whatever husbandry methods my brain throws out would work for them for getting a good colony going. I will be receiving various death feigners in large quantities in a couple of weeks, months, whenever the wild caught ones start becoming available. And I would really like to get those winterized individuals so I can, by my theories, get a large group of larvae going this summer of each, and then play around with mass pupation and whatnot, because I feel that's really the way to go for something like that, in my opinion. It's not very feasible or viable for me to have to separate out individual things. It's much easier to do groups for me. They eat super dry as larvae and eggs, or they drop like flies. Yeah, this sounds like something I'd be up for the challenge of. And having seen Vericosus in the as bulls Vericosus in the wild, um, it's the dryness and the specific conditions for different species are definitely a factor in where they can be found. So I think, is there anything else to touch on? Two things, I guess. Sometime this week, I will be cracking open the Goliath bin, and Kai has criticized me ruthlessly. No offense to Kai, I'm just simply bringing this up in case I need to be shamed or applauded when I check on my Goliaths. But he has criticized me ruthlessly for not providing dog food or something, a source of protein for my Goliath beetle larvae, saying that they will not mature unless they are provided some sort of protein source of that sort. So currently, I jumped the gun on, as I tend to do on things people who are listening may find out with some stuff. I jumped the gun on adding them to their new enclosure, and I did it without sort of having a game plan for where I was going to place handfuls of dog food or what have you. And I really just wanted to get them into a larger enclosure because they had been in a smaller one, and they were starting to consume up all the substrate. So I threw them into this new large attempted communal pupation enclosure with very high quality rotting leaves and good white rock colonized wood, etc. Um, and so this will be a check to see if the 12, I think I put 12 larvae in there. I still have a couple that I had kept aside to bring to a local reptile expo. I believe they're still in their cups. So, but anyways, this will be a good chance to see if they are doing, if Kai's wrong. But basically, bottom line, this is a good chance to see if Kai and I guess Alan are wrong with things here. So, and I do need to check and see if the pupation layer is of good quality. Although I made the damn setup so big, that I don't think I'll ever have to water it within the time frame of, the larvae pupating and closing as adults. There's an excellent degree of sort of moisture dynamics in that container, and so we'll see how things are when I check on them, because before they were temporarily in a 16-quart container, all 12 of them, third instars, with just high quality rotting leaf litter, and I think I put a layer of the white rotting wood on top of that, and they were all very healthy. And when I examined in the bottom, I noticed that they had sort of segregated themselves all into separate little sections. So no two of them were in direct contact. They were all sort of distributed throughout the substrate in a way that they weren't in contact with each other. Oh boy. The thing with the deregulation of the Goliaths, that's fascinating. And the other part of this is the whole, a lot of the things that I had read going into this, and that sort of like annoyed me with my experience with other sea needs, which is limited. I don't have any experience with Goliaths as of last year until I got them, but was the idea that they would only pupate in clay, and I was actually in sand. Oh well, actually no, not in this. You have to remove them to pupate. Blah 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 blah. All this other stuff that sort of went against what I felt was intuitive with cetanines. And then of course when I went looking for larvae to pull them from the enclosure, and I busted open a pupal cell that was made out of frass and leaf litter. And that adult ended up emerging with horrible wing deformities because I had broken the pupal cell and it was too far gone to fix it. That sort of made me question a lot of other techniques for raising Goliaths. So even if you are successful with that project, you might want to keep it to yourself. um, I mean, the thing is, the truth would get out eventually, and it's one of those situations where in that situation, if if people are sort of trying to assuming that this works out, that if people go around saying, "Oh, they need this. Oh, they really, really need this. Oh, they really. Oh, he's wrong," and I have evidence to the contrary, um, that's not that's not a good scenario, not just for me, but for other people who are looking to keep them. Because obviously, as I said before, when I was working with them, my goal was not necessarily to breed them, it would have been great, but I wanted to streamline the husbandry and get a better understanding for some of these things. Because obviously everybody has their techniques for stuff, and I have my tips and tricks for things. But getting away from the sort of superstition behind raising stuff like that, and sort of extrapolating from there, like the situation with the Ligia Palatii, where you have somebody who's selling something and they're like, "Oh, you just do this. It's very easy. Oh, you use this very simple thing. Oh, they really need this, or oh, the Cubars. Oh, you got to have limestone. They will all die without limestone." Where it's just misinformation that's going to waste people's money, and they're going to kill all kinds of bugs trying to do it this stupid way that doesn't work or isn't necessary. So, we will see what 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 I feel is the case, under the assumption that this does work and I get adults without having to give them protein. And I'm gonna make that call also when I check on them. If I open up that container, or if I go into that substrate and I find a bunch of fat, healthy larvae with a bunch of well-formed fecal pellets around them, and they're obviously thriving and doing well in this enclosure with the substrate and without adding the food, what I would think then is the cause of other people's failure is improper substrate, basically not providing them the right food that they need. Because with other seedlings that I have, like the Euphoria or the Gymnetus or the Pacnota or the Chlorocala, etc., a lot of them seem to be they they will eat rotting wood, but they seem to prefer to eat leaf litter. And people offering leaf litter to just about any organism, we've been doing it wrong for many years in terms of the state of decomposition that material needs to be in, and they want that stuff that is just primed for turning into soil, basically. And when you collect things, find seedlings and other beetle larvae in the wild in leaf litter, that's the state they're in. They're in that nice humusy layer of leaf litter that is just in very accessible nutrition wise to them, because of the state of decay that it's in terms of colonization with fungi and microbes. And it's either actively decaying or it has already broken down to a point where they don't have to worry about digesting the cellulose and whatnot that's in it. So somebody who has more experience raising things like lecanids or dynastines may approach Goliaths thinking that they need to eat, mostly a woody material, etc. And then when the larvae do not do excellently on that diet, thinking, oh, they need more protein, so throwing in more protein, dog food or fish pellets or whatever, and then they start doing well because they have something that's more nutritious for them directly on hand and available, would think, oh, they require this, when in fact, no, they require a different substrate material to thrive without supplementing like that. So that's what I'm trying to piece together and find out here. And it's a large container. It's probably it's a little bit smaller than the desk that I'm at here. It's a little bit smaller than that, and it's got maybe like 5 to 6 6 inches of the clay sand at the bottom, and then of course all the rich rotting leaf litter, along with the actively white rotting wood pellets on top of that slash mixed throughout that. So supposedly have adaptation to hunt other insects, if I remember correctly. I would be interested in seeing uh, a solid documentation of that because I did sweat a little putting them into that 16 quart temporarily because it's like, Kyle, you're dumbass. Why are you doing this? You should be putting them into the full on setup, but it wasn't ready yet, and I didn't want to have to extract all the water from the clay. I wanted it to dry out as naturally as possible because it's a lot of work and a lot of mess to extract the water from it. So, but then looking at the bottom and seeing the way they had distributed themselves and the fact they were actively eating the leaf litter and seemed quite plump gave me a bit more hope and galvanization in what I was thinking. So, and another thing might be space. They are large beetle larvae. They need space so they don't have to constantly be in contact with each other, which may be more important than providing the extra protein or whatever for them. Because if they can segregate by space in this enclosure, they can continue munching away at their pockets of material without ever having to come into antagonistic encounters. So, not to completely derail the combo here, but found 9 as Bullus mexicanus eggs right now. That's wonderful, and they have it labeled to subspecies as well. Yeah, I'm looking forward to throwing my hat in the ring with the Asbolus. I'm not exactly. No, I know why I didn't do it last year. Probably because I didn't have the funds allocated to buy Asbolus, and I was also going to be busy all summer with the plant work, which won't be the situation here. They can be reared on decaying organic matter until they reach L3. From what I've heard, L3 won't grow without protein source. Well, we will see, and I continue to say this, but we'll see what happens when I check on them because I received most of them as late L2s or early L3s. So we'll see how much larger or smaller they are when I check on them in a couple of days. And I really loaded up that bin with quality stuff. So, and it's the white rotwood was actually so actively colonized with fungus, it's actually growing pleurotus mushrooms right now. And the other thing is the mycorrhizae, or not mycorrhizae, the mycelium from the white rot. The mushrooms are proteinaceous. People on vegan diets and whatnot use them as a protein source. So, it wouldn't be entirely fair, I guess, to say that I'm raising them without protein because, there's all the mycelium that's mixed in there and colonized throughout the wood. But it's certainly not, a handful of dog food or what have you. So, so yeah, there was that. I feel like there was one more thing I was going to bring up. Goliaths. Let me take a look here and see if there's anything else. No, don't really think so. I covered a lot of ground today, so any beetle things maybe. Dynasties are all doing well on my compost. I actually swapped Kai for some Dynasties Grantii, not Grantii, as has been hammered in my head a couple times now. Dynasties Grantii, and if I have a good picture of an adult male, I will probably add Dynasties Grantii to the website. At least because if I can't continue propagating them myself, I know plenty of people who can, who would love to sell me larvae wholesale. So Dynasties Grantii may be added to the site, so I'll have both Titius and Grantii up for sale on there. So anything else? Anything else at all? Did I go downstairs and just say wow at something in the last couple of days? Gotta be something that I looked into and was very happy about. something that I maybe just started, maybe something up here. I regret putting Latiblatella renai and Neoblatella detersa in the same enclosure because I realized that it's very difficult to tell the small nymphs apart, and I have no idea, or I'm going to have no idea, if I'm seeing second or third instar Neoblatella detersa or Latiblatella renai. So I'm just going to have to suffer. Panchlora species white update. All right, just for you, TJ. I found, I think they arrived with the material that they were shipped in, but I found planaria in with them. I haven't done a head count on them, but there was one larger nymph that had sort of a um, uh, wing pad deformity. It was like swollen with hemolymph. It didn't seem like the prognosis was the best. It looked like it might mature, but with a severe deformity on one wing. The last time I checked, I don't think I saw that nymph. And of course, now noticing these planaria that have popped up in there, I actually have the container on my workstation, and I'm going to be moving them to a new container tonight, so that we don't have to worry about any of this planaria nonsense that could be causing issues. From the rack. Euthlastoblata species Ocala. Yeah, really quick though, Alan. Yeah, that's easy to say, but not when I go to lift a piece of bark and a billion of them scatter in every direction, and I'm not going to have the patience to go after them with my pooter to separate them. So I have resigned myself to waiting until there are more adults or sub-adults so that I can be absolutely certain, and then I will be pulling them into their respective future containers. Uh, so yeah, so those are going. Simplos morsi. There's a bunch of oots. Haven't seen babies hatch yet, but I'm taking good care of them now. And so I think this will be the time I finally get them established. What I was trying to say was the Eulastoblata species Ocala have finally started hatching, and I think I mentioned last time that the species from the Everglades area actually was able to um, lay oots, and the oots hatch in a shorter amount of time overall than the Ocalas were laying oots and then having them hatch. So there is, even under the same or very extremely similar conditions, there is a difference in hatching time between the Ocalas and the Everglades, which I feel is a strong argument for them being something different other than the fact that they're morphologically different. So so yeah, there are Ocala babies or newborns, and they're popping up. Most of the adults that emerged are still alive and doing well. So I'm imagining about a month from now, I will be just drowning in them. So TJ, if we continue to postpone me sending you some bugs, I can include some Ocalas with them, but I don't feel comfortable shipping them at their current size. I would much rather wait until they're L3 or L4, and they'll also be easier for me to catch and to pack up a good number at that size. So currently for Euthlastoblata species, I have a very hardy culture of Diaphana. We have species Everglades, which are hatching. The adults are still alive, so we're going to have species Everglades, whatever they are, which I have a suspicion they're Diaphana. And then we have species Ocala, which seem to be something completely different. So I feel pretty confident that I'm going to be swimming in Ocalas. So I'm probably going to add them with the Roach dump. And for common name, because they're probably undescribed, although we do have to do a bunch of literature perusing, etc., before we make any definitive conclusions. I'm very, very tempted to give them the common name because they're undescribed. We don't know what species they are, although if they have a species name, we figure that out, determine they're probably a different species. You can always change the common name. but I feel like the name Florida Beauty Roach is very applicable to them because the nymphs are, especially after seeing that one in the wild and just being absolutely mesmerized by it, I feel like it's a very good common name. And even though the adults are not as pretty as the nymphs, they still have something going on for them. So probably put them up as Florida Beauty Roach. And really, the only people who would care or object to a possible new common name are in the chat right now. So if Florida Beauty Roach does not seem applicable to any of you guys, speak now or forever hold your peace. You have 1 minute to respond because I just heard Victor whine, so he probably wants some attention. And we are past the 2-hour point. So, Eulastoblata species Ocala, common name Florida Beauty Roach. All opposed? Or all four? All four, all opposed? Florida Gem Roach. I mean, you already have a Gem Roach that's a Glauterix gemma is almost endemic to Florida. I think it's found in some Caribbean islands, but it's check the bug guide, and it's not that bad. There's not bug guide, the INAT. Let's see. But I would assume the epicenter for the rain. Oh, it's a southeastern United States endemic, but you would never think it, considering we always find them in Florida, or rather, Alan always finds them in Florida. So, yeah, so it's a southeastern endemic, but I mean, most of the time that we get stock, it's from Florida. So, Alan is 2 against 1 there. We'll see if Joshua Campos is still here, if he has any say, or if anybody else has any say. But I feel like it just we don't have any records from anywhere else in the United States, or even any bug guide or INAT things or something that looks like that. So, but anyways, I guess we can always bring this up again. It's not Sunday anyways. So, with that being said, I'm going to take off. Excellent stream, everyone. Remember, next week's stream, which I will be changing the name of the stream for that particular stream, just so that everybody knows it's going to be something a little different. Oh, you did? Did you find the? Oh, you did. Do you think they were the species Ocala or the Diaphana? Alan says he found Eulastoblatus in Alabama. Anyways, we can talk about this again. I really should get moving on some other stuff. So, but anyways, next week's stream will be a more focused topic. Obviously, I'll allow questions in the chat. I'm not going to tell somebody to shut up if they have a good question about something completely unrelated. But I would really like to focus on this one particular thing because of my the importance I feel that they have. Alan, we'll discuss this in a little bit or another day or something. So before I go slapping names on things. So, anyways, thank you all for joining me once again for this wonderful, I feel like this is a pretty quirky and very well-covered stream. With that being said, I'll see everyone at the special stream next week. And until then, may your cultures be ever bountiful. I'm trying to make this my sign-off thing, but I keep screwing it up. May your cultures, may your colonies be ever bountiful. May your cultures be ever bountiful. I don't know, one or the other. So, anyways, good night, everybody. Thanks for joining me, and see you all next week, hopefully.